Pests of sugarcane: shoot borer, white flat eggs overlapping each other in clusters containing 80 to 60 eggs in 3 to 5 rows on the underside of leaf sheaths. Larvae hatch out from eggs in 2 to 6 days. Enter stem by making hole. Have 5 violet strips dorsally and dorsolaterally on its body, and its head is brown. Pupa pupate inside the stem. Adult moth emerges in 60 to 8 days. All pale grayish brown moth with white hind wings. Damage: the insect is widely distributed and attacks 1 to 3 month old crop of sugarcane. The larva tunnels into the stem, causing dead heart, and 10 to 15% of canes are damaged. Occurs during pre-monsoon period, March July. Its multiplication is favored by high temperature and low humidity. Control: cultural method. Time of planting: early planting during December January to escape the shoot border incidents. Trash mulching and earthing up for early shoot borer. Removal: destruction of infected cane. Irrigation of closer intervals for managing early shoot borer. Removal of water shoots to destroy top shoot borer. Cultivation of resistant variety: CO975, CO46, CO7004, 7304. Biological control: Trichogramma caelonis for early shoot borer. Chemical methods: chloropyrifos 4 applied by soil drenching. Then spermethrin fipronil and can be sprayed. Stem borer: scale-like eggs in batches, each batch containing 9 to 11 eggs in 2 rows. On the sheathing leaves, maximum of 400 eggs are laid by female. Incubation period 5 to 6 days. Larva bows at the nodal region and enters the stem. May attack 1 to 3 internodes. White body with dark spots and brown head become full grown in 37 to 53 days. Pupa: larva pupates in the leaf sheath. Pupal period lasts for 8 to 10 days. Total life cycle 50 to 70. Adult moth is pale brown with white hind wings. Damage: the insect usually occurs on sugarcane late in its growing phase during June and December. Multiplication: rapid stem and its feeding causes the tissues turn red and the hole is usually plugged with excreta. Its attacks significantly affect juice quality if damage extends to 3% or more internodes for more than 10% of total length of cane. Then reddening of affected tissues inside the cane. Control: no effective chemical control measures. Periodic collection and destruction of egg masses may be useful. Release of egg parasitoid Trichogramma caelonis. Top borer: dull white elongate overlapping eggs in clusters covered with buff colored naves from female anal tuft. Larva: dull creamy yellow larva about 25 mm long, become grown in 37 to 53 days and pupates inside the stem. Pupal period lasts for 8 to 10 days. Adult whitish in color with buff colored anal tuft at crimson colored tip of the abdomen in gravid female. Damage: it occurs in sugarcane in the later stages of growth during June, December and may persist till harvest. Parallel rows of short holes in the emerging leaves causes a white streak which later turns reddish brown. Dead heart in ground up Cans reddish brown in color which cannot be easily pulled. In tillering phase of crop, attacking shoots die, side shoots develop producing a bungee top appearance. Control cultural method, use of pheromone traps, 5 per acre for monitoring of adult moths, collection of destruction of eggs. Biological method using Trichogramma cheilonis or Isotina javensis. Chemical method, carduran 3G or Forate. Black winged bug. Adults are black in color with white patches on body. Young ones are similar to adults but smaller in size. Damage affected crops become brown and red spots appear on the leaf. Pest active throughout the year but most active in the dry and hot season when there is no rain. Control frequent irrigation reduces attack. Avoid use of excessive nitrogenous fertilizers. Burn stubbles to control this pest. Apply following granular pest insecticides. Imidacloprid 5-Pronil. If spray is needed, spray with fibronate. Sugarcane leaf hopper. Yellowish eggs in groups, each containing 10 to 58 eggs. Nymph covered with wax and has a pair of long and wax covered oral process. Nymphal period lasts for 50 to 60 days. Adult brown, densely veined, transparent wings having pointed snout. Damage both adult and nymph. Suck the sap from leaves. Attacked leaves get folded and dried up. Plant shows sticky appearance. Then mold develops on leaves due to honeydew secretions of bugs. Control, collect and destroy leafhopper. Use contact poison BHC and malathion 0.1% are quite effective in controlling the pest. Endosulfan 0.07% is quite effective. Sugarcane scale. Melana. Sugarcane scale, grayish black or brown circular scales that cover the nodal region following thick encrustation as the whole stem. Life history, reproduce ovoviviparously. It has 5 larvae instars. Lifespan of female is 34 to 62 days. Sex determination take place in third instar. Pupal period of male is 60 to 13 days. Total lifespan 19 to 36 days. Damage, grayish black circular scales seen covering the nodal region and form a thick encrustation on the whole stem. The nymphs and adult female inside this scale covering feed by sucking the juice and cause shriveling up and stunting of canes. Such canes will slit open, appear brownish red. Control, stripping cane leaves may produce attack. Use scale-free stems for implantations. Spray malathion or dimethoate. Soil applications of carbofuran at 2 kg. Use of hymenopteran parasitoids like Anabrotepsis mayuri sp. Termites. The infestation commences with panting of sets and damage the cut ends and bud sets affected their germination to the extent of 28 to 68%, resulting gaps in field sets are completely eaten away, leaning only the outer hard riots in the attacked plants. Outer leaves show drying canes may come off easily if pulled. Lateral parts of malleable canes are eaten, results in heavy loss in yield. Crop raised in light soil suffers to greater extent under drought condition. Control, lindane emulsion application on sets and furrows at the time of planting selected for shoot border control is effective in termite damage also. White grapes. The eggs are oval creamy white with when fresh and later turn to brown in color. Grub, the newly hatched grub in white and scarabaeiform pupa. The pupa is exarrate type and pupation take place in soil. Generally adult or lamellate and males being smaller than females. Dark brown beetles. Symptoms of damage. Both grub and adult cause the damage. Grub feeds on fine rootlets and then girdles of the main roots. Yellowing and wilting of leaves and finally dies out. Affected canes come off easily when pulled. Control cultural method. Collection of destruction of adult beetles from host trees. Picking up grubs manually from field. Repeated plucking and exposing various stages of grub to the natural enemies. The use of light trap. Biological method. Bacillus bacteria and Brevia bassiana. Fungi can be used. Chemical method. Adult can be controlled by carbaryl 0.1%. Monocrotophos 0.05%. Chloropyrifos 0.05%. The grub can be controlled by Forate 10 gram at 25 kg per hour. Then mealybug Pseudococcidae hemiptera, pinkish oval insects beneath the leaf sheath and on the notes with whitish waxy coating. Mealybug. Symptoms of damage. Nymph and adult suck plants juice from canine group and induce vigor to the reduced vigor of the plant. It also attack roots. Honeydew secretion leads to development of sooty mold which gives blackish appearance to canes. Control. Use resistant varieties like CO439, CO443, CO720, CO730. Drain excess water from the field. Detrash the crop on 150 and 250. After planting, apply methyl parathion. Drain excess water from the field. Detrash the crops on 150 and 210 after planting. Apply methyl parathion. Rubbing of sets with bag pieces stripped malathion. Stripping of leaves of spraying parathion 0.05%. Then white flies. Eggs fixed on leaf tissue on both surfaces. Incubation period varies 9 to 13 days. Nymph oval black with fringes of wax and waxy deposit on body. Adult pale yellow, adult female. Life cycle 32 to 44 days. Damage dense white line infestation may result in reduction in cane yield up to 24% and loss in sugar up to 2.5 unit. Control. Discourage rationing in low-lying areas and avoid waterlogging. Remove lower leaves containing pupae periodically. Avoid use of nitrogen fertilizers. Spray endosulfan 0.01% or malathion 0.1%. Nymphal parasitoids and predators can be used.